being able to do research here, even as a freshman, to be able to ask to do research is awesome.
The students have been, they initially isolated soil samples from, some of them took them from campus, some of them from their homes, whatever, and then they took those soil samples and mixed them with bacteria and then separated the bacteria from viruses that infect those bacteria. So it's the viruses that we're actually interested in. And so then they purified those viruses and so that they could come up with a single species of virus, and then spent the rest of the semester characterizing that virus. They took pictures with the electron microscope over at Boise State, which was really fun, and then they isolated the DNA and did a little bit of work with the DNA, and then we chose one phage, one virus from the class to send away to have the whole genome sequenced.
I think one of the things that I think we're most excited about for this course is that they all complete a research project through the course of this semester rather than doing a lab where they do one lab exercise one week and they do another lab exercise another week and another lab the next week that don't necessarily relate to each other. And they might illustrate some concepts from the lecture, but I think this gives them a much more realistic exposure to how science is actually done.
Several schools across the country have done this, and no one's come up with the same virus twice. So presumably every student has their own unique phage. They've given it a name. They've put it on a national database.
If someone wanted to look at, say, phages that were found in one part of the country versus here in Idaho versus another part of the country, they could pull samples off the shelf and they'd be able to do that comparison.
This was the first major, long research project I've ever been a part of. I learned that it takes a lot of time and work, and you make more mistakes than you think you'll make.
It's essentially a collaborative project across the United States, and I thought that was really cool that, you know, a small school gets to do a huge research project.
Well, we named our phage Spunk because there were several times throughout the experiment where we'd make little mistakes and we thought that we were going to have to redo a step or redo a test, but our phage was pretty strong, kept on going.
With their own sample and a whole genome that they've actually discovered for themselves, rather than just kind of looking online at some database and selecting some organism that somebody else sequenced. I think they get a lot more ownership out of it.
One of the greatest things that they've learned as far as being in the lab is that they really became comfortable with the techniques and they became comfortable making decisions.